for asking me to give a talk on genomics and genetics in prostate cancer. Why a urologist or a prostate cancer doctor should know about genomics. I'll give you my background, I'll give you my understanding, but very early on I got to comprehend that not everything in prostate cancer I can treat just by the knife, and in my case, with the robot. With that understanding, I started investigating more in depth about what is happening in the genomic level, and I will give you summary of the journey which I have taken. What we need to understand that I'm in front of you and I am some total of about 30 plus trillion cells. My body is made up of at least 30 trillion cells, and many of these cells every minute are dying. Not only they are dying, they are being recycled back. So this process of my cells staying in a constant lively stage, some of them dying and some of them are coming back. This entire process is a very complicated process, and what you are seeing on the screen on the right side is a cell which is a very complicated structure in itself, and as it evolves into from one cell to this 30 plus trillion cells, it differentiates into different kinds of bodily organs. Some of the cells become the blood cells. Some of the cells become the eye cells, some kidney, some skin, some liver, and some prostate. These cells, they started from an original one sperm and one ovum and evolved into a trillion cell factory in which there is a social system which exists, in which one cell chooses to float around in the body carrying oxygen, the other cell works on excreting waste products from the kidney, and there is another cell which is a metabolic factory in the body and makes the liver. And then there is a skin, there are body's defense mechanisms, there is a nervous system, there are bones, there are accessory organs. All that has evolved due to a very controlled differentiation process which our body, our species has learned over millions of years. But this process is not perfect, meaning whenever there's this new cycling of the cells which is going on, they generate back into its normal form, but sometimes there are mistakes which happen. And those mistakes can actually result in cancer. And that's the reason, as a surgeon, as a scientist, I have to learn about what is happening at the basic molecular level which can impact the whole bodily function. And the other side of the screen is showing something what we call cell cycle process. Most of the cells, they are staying dormant, meaning they are doing their job, but they have no desire to multiply. But there are certain cells in the skin and certain cells in the lining of our gut, the certain cells in the bone marrow. They have to multiply again back because they are having a issue with the cell death, and the cell regeneration needs to happen. And this entire process of a resting stage versus what we call a G1 and a G2 stages, these are the cell cycles which is very delicately controlled. Not every cell can go on to a synthetic phase or go on to a G1 to the G2 phase or go on to the mitotic phase. There are certain checkpoints which exist, and these checkpoints are at 3 different levels, and these are some kinds of proteins which actually control the entire process. It checks on whether there is enough organelles present before the cell is ready to divide. There is enough DNA made and duplicated before the cell is ready to divide. Has the DNA duplication Has that process been taken through the quality checks or not? Because we don't want to create a cell which is not the perfect form. That entire built in check and balance mechanism is what we call our life. And understanding about that life is the journey I'm taking you through the cancer genetics discussion. If I have to sum up why are we having this discussion as a surgeon, as a scientist, it can give us an insight as to why prostate cancer happens. Genomics will help us in understanding who is at risk, how we can better diagnose prostate cancer. We have a diagnosis of prostate cancer and we can use genomic tools to make a better prediction as to which amongst these 10, 20 patients is likely to be a very advanced growing cancer versus one which will be very indolent. Genomics can tell us. We can manage the recurrence better just because we know what kind of a cancer has come back. And today, we are able to develop certain treatments which are targeted towards a particular genomic subtype. And as the end of this discussion, you will see we can even devise body's own defense mechanism to fight against prostate cancer if we understand the genetic detail of that specific cancer. What we call personalized genomic vaccines can be made. So, genomics is not just an intellectual exercise, it's going to help us be a better doctor, better surgeon, and a better clinician. Let's go through this journey. I'm showing you on the left side what I get to see as a surgeon. As a surgeon, I see a structure, I see an organ, I see facial planes, I see tissue. I can understand where one structure ends and the other structure starts. On the right side, what you are seeing is a gross specimen which has come out, and that is the gross pathology, which is telling me that I have taken the organ out, and part of that organ is a diseased, what we call cancerous in this case. On the lower left side, what you're seeing is a zoomed in mechanism. First I saw the structure, now I'm seeing a gross image, and now I'm getting deep inside under a microscope. By magnification, I can see structures, that where the cancer is, how cancer is trying to escape, and if cancer has become from an incurable to a curable to an incurable cancer because it has broken through the anatomical boundaries. On the right side, what I'm trying to suggest that all that is happening in an organ, in a tissue, in a microscopy actually is driven deep down inside the cells by certain proteins. Mind it, those proteins are making a cell become from a normal to a rogue cell. And that understanding of those proteins, those pathways, those molecular pathways is where the genomics comes in. So, with this background, I want to show you a first ever case of an prostate cancer which was diagnosed in about 2700 years ago. And that happened to be a king of an area which was between the Russia and Afghanistan, and that was known as the king of Azran. That king was very strong person, but was the first case of documented prostate cancer in which we have been able to confirm that because of no other method but for the genomics that allowed us to confirm that he had a prostate cancer. What you are seeing in the bottom is the journey which happened in that patient from a single cell to a clump of cells to a more invasive pathway, and cancer became totally out of control. And on the right upper side, what you're seeing are the bones which became brittle, eaten away by the cancer, and that finally killed that king. Strong person, prostate cancer started in a cell, started in an organ, grew beyond the organ, and overtook the whole body. That all can happen, and I want you to take through this journey of going inside the human body What you're seeing now is in bladder, then you are seeing a prostate, and in the prostate, what you are seeing is one rogue cell decides to become two, two becomes four, four becomes 16, and then they grow to become millions and sometimes billion cells. Up to this point, it's all okay, but then it gets to a point where it reaches the blood vessel, reaches a nerve, and gets out of the boundaries of the organ where it actually took the origin from. And now it is becoming a problematic cancer. It is becoming what we call an invasive cancer, and then it will become a regional cancer, and then finally it will become a metastatic cancer, which will go to any part of the body. It went to the bones just like it went in that King of the Azran. How that single cell evolved from a very normal prostate cell into a rogue cancer cell became so angry that it took over, broke through all the control mechanisms, and then went on to overrule the whole body, and in that process killing the human. That all was happening because of certain changes which were happening inside the cells. And as a scientist, we can understand those changes, and that is what we call the genomic details of the cancer biology, which can have an impact on the final outcome, because there are certain treatments which may be there if we understand what exactly is happening inside the gene which is abnormal and can be fixed. That's the reason we need to understand the genomics, and I want to bring you back into the discussions about the proteins. Everything which happens, you are watching this video, and this video is being watched by your eyes, and there is a protein in the retina known as rhodopsin, and that is changing the chemical structure and creating light into a neural impulse. It's going inside through your nerves, inside your brain, and the neurotransmitters are acting on it. The very fact that I can speak, I can move my hand, it's because of certain muscle proteins are acting and allowing my voice to come out, my fingers to move around, and I can change my gesture to look at it. All that is happening because of proteins inside the cells. Those 30 plus trillion cells I talked about, they have proteins inside. And the proteins are very, very important part of our body, because not only that, they can allow us to see or hear, but they make the structure of the cell. They act as a messenger of the cell. They are involved as an enzymes. They are involved in the cell communications. One cell talking to the other cell, hey, oxygen has arrived, or an invader is there. All that messaging system is happening through the enzymes or through the growth factors or through the signals. They're all made up of different forms of proteins. Those proteins can also be very helpful for us to build up an immune mechanism so that it can find and fight an invading bacteria or invading COVID virus. All that is being managed and orchestrated by proteins. So proteins are our acting chemicals. Guess how these proteins are made? That is where the gene discussion comes in, because proteins are not only involved in the day-to-day functioning. As I said, there are certain checkpoint inhibitors. The process of entire cellular division itself is controlled by the proteins, either as a structure or as a messenger. And this entire process is controlled or coded by the genes. And that's what we need to understand, that each one of us have two books of codes given to us, one by my father and one by my mother. These two books have the entire genetic code written to us so that we can read it and we can copy it, and then we can create a specific protein whenever and wherever it is needed. Each one of these books has 23 chapters. 23 chromosomes. These 23 chromosomes are coming from father's side and from the mother's side, but these chapters have got small paragraphs. These paragraphs focus on one particular recipe. Let's look at the recipe is we want to fight a virus, we have a different kind of chemicals we need to produce. We have little lack of oxygen because we have gone to a higher altitude and we need to get in more RBCs in our body. A different other kind of protein needs to be produced. This all recipe in a very perfect form is written in our genetic book. This is our genome, and the alphabets in which these paragraphs and chapters are written is known as language of nature, language of life, or what we call a DNA. Each one of these chromosomes is a very complexly folded form of an DNA that is wrapped around, and I'll come on to as to how this wrapping even can have an impact on how things are functioning. But this DNA has segments. These segments are what we call genes. A particular paragraph of a particular chapter talks about a specific functioning, and these are what we call genes. And guess how many genes we have? We may have more than 20,000 genes, but at least these 20,000 we know of. And these 20,000 genes are coded or written by the DNA which we have. And the intermediary of this DNA is the book. We have a scanner or a reader, which is an RNA, 3 different kinds. They get the job done inside the nucleus, get the RNA out, and go and produce protein, our favorite protein. And that entire process is very complicated, and because DNA message through RNA is making a protein, but there are only 20 different kinds of amino acids. And these amino acids can be recycled, repurposed, refolded, recombined, and then shapes change to 20,000 of the proteins. All that is happening inside those small cells of ours. And this entire process is a controlled process. So that's the reason we wanted to understand how this process is being controlled. Book exists, but we don't take care of the book. The DNA messaging gets a little bit distorted. Message was there, but the reading didn't happen very well. So what we call a mutation happened. A particular segment of a gene was either a misread or over amplified, or chromosome because of some reason got shattered and then got recombined, and the net result was there was a distortion in the actual true meaning of that gene, and that's exactly what we call mutation. Mutations happen every time. Mutations, they happen in our body all the time, but many of those mutations are not meaningful. Some of the mutations are meaningful, what we call the driver mutations, and some of the mutations are not compatible with the life. So what's happening while mutations are happening? There are different kinds of mutations can happen. You read it wrong, or when you typed it wrong. It's like in jumbled up typewriter in which the two alphabets are writing a wrong message. So this is a single alphabet is being transcribed wrong. But if a small segment is the problem, that entire thing will produce a protein, but instead of producing a squash, it will add sugar to salt. And guess what? That drink is not going to be very useful because it was supposed to be sweet, but somewhere the letters got mixed up. And if that happens in a very relevant gene, which is producing a protein which is critical with the life of that cell, we cannot let that cell go on. and that is where the control mechanisms they come. Body has a very good control mechanism. Not only it's doing a proofreading more than once, it is also looking at the final product. And if the final product has a problem, it has to make a decision. Is this cell fixable? We have a mechanism known as the DNA repair pathways. Or is this cell not compatible? It's so defective that if we continue it living, it will create problems. Then that cell has to die, and we steer that cell towards what we call apoptosis. That cell decides, you know what, I live my life, I have a defect, and if I don't commit suicide, I will be damaging my neighbors. So this is a social system built in our millions of years of evolution, and this entire process is very well controlled. So what we learned, proteins were important, genes are in the chromosomes through the DNA, mutations can happen, but body has a very decent safeguard mechanism of dealing with these mutations. So I started getting more and more involved in this gene and how things are happening. So genes are the one which are controlling the proteins, and one cell to the trillions of cells. There is a total art in how this entire DNA is packaged. By the way, I mean, it's a more of a trivia question, and basically each one of us have enough DNA folded very meticulously inside our cells that if we unfold it, we can go from here to sun 300 times. Enough length of a DNA exists in each one of us that if we unfold it, that distance will be from here to sun, not just one time, 300 times. So basically, there is a lot of information is kind of arranged and coded within our DNA. And every now and then, DNA having a special protein around it, what we call histones. These histones, they allow you to kind of do the packaging in a way so that it is compressed within a very small segment. And sometimes the proteins around the DNA also make sure that not everything which is out there in the DNA is coded. So this is a very precise machinery, and we as a surgeon scientist, and now you all have to comprehend it. So I'm trying to simplify it as much as I can so that you get the bigger picture. Obviously there are textbooks and books for us to get into the deeper dive into this. So DNA is an important part. Nucleosomes, histones are very important part of this entire factory. There is a lot of DNA in our body, and there is a lot of methods in which this entire thing is controlled. I also want you to know, without getting into the too much of a detail, it's like very early on in our career we were taught how to read the EKGs, and we were given the stethoscopes to hear the heart murmurs and the sounds and all the signs and symptoms of the entire general exam. Same way a new field has come up, and there are different ways of looking at the gene. There is a phenomenon known as a whole genome sequencing. There is an exome sequencing. There are even what you call a methylation for the histone sequencing, and there are ways to get into the single cell and what we call a single cell sequencing. So we know that the DNA has genes. Genes control protein. Protein is important for this timely and regulated duplication of the cell, which is the reality of life. Every body has cells which are dying, and some cells need to come up. In that process, certain cells misbehave, and when they misbehave, they become cancerous. And my next transition point is, what do we mean when things become cancerous? And I want to simplify it. There is a very important paper which has been written about the cancer, and cancer cells share certain basic tenets why they become cancerous, and these are known as hallmarks of cancer. There is a very complicated diagram on the left side in which we talk about that there is a phenomena of overcoming the growth suppressors. There is a problem with the immune evasion. But I will simplify this entire process into, imagine you are having a car ride. You are driving, and at a curve, you are supposed to take your foot off the pedal so that the accelerator slows down. Suddenly, you realize that actually the car's accelerator is stuck. If that happens, car will not slow down and will keep going. Same thing is happening in a cancer cell. Somehow cancer cells were supposed to slow down in a division process because there were enough number of cells, and certain accelerators are stuck, and we call them oncogenes. Oncogenes are certain proteins, certain genes, certain molecular pathways within the cell which just keep telling the cell divide, 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 divide. That should have slowed down, but something has happened to that cell that it is not slowing down. Okay, you cannot take the accelerator off, but you can, in that situation, apply a brake. And you apply a brake, and then suddenly you realize that the brakes are also not working. What I mean, that in a normal cell, when there is a hyper accelerated pathway which is active, there are certain brakes which are built in, and these brakes are known as tumor suppressors. Tumor suppressors can block the entire pathway and then say, hey, you know what? You need to slow down. But cancer cells somehow figure out how to even override that control, and what is happening that these cells, they don't stop. So on one side there is an oncogene which is driving the mutations, driving the cell growth, and the other side the normal control mechanism is also not working. What we call the tumor suppressors are not working. I'll give an example of an oncogene. Is a RAS is an oncogene, Pi3 kinase pathway is another one which is involved. The P10, a P53 are what we call irregular tumor suppressors. Then comes the third thing, that okay, sooner or later if you keep dividing, the normal cells have a limit known as limit of about 60 to 65 cell division, after which cell runs out of its dividing ability, and then that is controlled by something known as telomeres. Telomeres at the end of the chromosome, and if you don't take care of it, it frays and the DNA itself cannot divide, and it's known as a Hayflick's limit. That limit is there because of shortening of the telomeres, and we hope that the cancer cell after X amount of divisions will run out of the telomere and it will stop dividing. Now, cancer cell has figured out how to rebuild the telomeres. So the telomerase enzyme is there, and that enzymes keep regenerating back that portion. So multiple things are going on, and then you hope that it will run out of the gas. The carburetors are problematic and it will run out of the gas. So what we call the normal energetics of a cell is that the mitochondria works under aerobic conditions, and the aerobic conditions produces certain ATP, and cell needs those ATPs in a very efficient manner. Cancer cells have hacked the carburetor, and they can work in a non-aerobic pathways, and they produce more lactate and all those things, and that lactate is used for some other kind of an structural phenomena, and they have rigged the whole carburetor system. So this entire process is happening inside a cancer cell, and we need to understand what's happening and what is driving at the genetic level. Then think about, you are driving this car and it is just going fast and fast and fast through the road, not respecting any kind of control mechanism. You hope that sooner or later the traffic police will get to you and will finally force you or somehow manage to stop it. Yeah, that's supposed to happen to the normal cell, but the cancer cells have figured out how to not be seen by the traffic cops. So our immune cells are supposed to stop it, but the tumor cells have figured out how to hide their antigens so that they are not exposed and they can go on for a long time. Guess what? This all is happening in a cancer cell which has just refused to follow the social rules of not dividing beyond a certain number. Next thing comes that the normal cells will run out of gas, meaning they need oxygen, and after a while, when the number of cell has grown beyond a certain limit, it will be needing more oxygen, but there is no more oxygen to come. But that's where the cancer cells become a little tricky and conniving. They will produce what they call tumor angiogenic factors. So they will create chemicals which will create sprouting of the capillaries so that they get a preferential blood supply of more oxygen coming to them, and that phenomena is known as angiogenesis. That all is happening because of certain enzymes, certain proteins, certain genes are acting and helping cancer cell in getting all this achieved. Few other things are going on. One of them is that the gene itself in that cancer cell is not very stable, and we call it genomic instability happening. Other thing is, when there's so much of an unregulated cancer growth is happening, the body produces some inflammation, and inflammation now colludes with the cancer and making it even more aggressive. So pro-cancerous inflammatory process is one of the hallmarks of the cancer. But to me, one of the biggest tools which cancer cells they use to become from in one solitary cell to a billion cell mass is what we call this vehicle, which was only trained to drive on an highway, on a road, changes from a regular cell to what we call an all-terrain vehicle. What is happening? The cell which started early on a de-differentiated cell has become differentiated to be a prostatic cell, which is supposed to be not move around. It's supposed to sit on an basement membrane, produce certain chemicals and the PSAs and all those things. This cell is regressing backwards to a point that this regression will result in what we call an epithelial to mesenchymal transformation. This car is now becoming an all-terrain vehicle. It can grow within the collagen network outside into the tumor microenvironment. It can drill holes using the inflammatory cells and other methods and metalloporphyrins to go outside the capsule. It will collude with the nerves to find its way out. It will then go to the lymph nodes, it will go to the blood vessel, and now it has become a metastatic niche. It's circulating everywhere, and finally, sooner or later, it will find a new home, will produce this entire process one more time, and this is what is the journey of the cancer. And this complicated slide in front of you summarizes that many different proteins, many different genes, many different pathways are what are doing this entire damage to the human body. Okay, it was a little complicated, but we had to go through this because in order for me to explain, I myself went through this entire process, and it all started when I started collecting tissue during my robotic surgery to understand what is happening deep inside the cell. I started taking tissue from the different parts of the prostate. I started doing a single cell genomics, and I started looking at the what we call the copy number variations, and even in one different location, one prostate, one cancer, different parts of the cancer were different clones. That resulted me in understanding that this is in clonal evolution which is happening. That brings me to a different thought, what I'm going to introduce, that prostate cancer carries name prostate cancer in 10 different patients. You think they may have a different stage, they may have a different Gleason grade, but at a fundamental level, at a molecule level, at a gene level, at a protein level, at a pathway level, these 10 cancers will be very, very different. And I'll get to that why it is important, but cancer may become a very aggressive cancer taking a totally different pathway. And that is what is what is being shown here. It's one of the first papers which I wrote with my colleagues in Cornell and at Broad Institute in Harvard system. These are all my patients. They have different Gleason grades, but when you look inside, what you are seeing here is known as circogram. Circogram is an EKG for the genes. Each one of those numbers represent one chromosome, and it is also showing whether a part of it is deleted, added, translocated, and if it has translocated, where it has gone and got attached. This is what the circogram is, and just looking at it grossly, look at the right lower one and the left upper one. There are two different kinds of circograms, and that's exactly what is happening at the genomic level. 7 different patients, 7 different genomic types. So this phenomena is known as an inter-tumor heterogeneity, meaning between these different patients, don't expect them to have the same kind of cancer. And we started looking at what exactly was going on, and these are some of the papers we wrote as to what is happening at the mutation level and what was happening in the epigenetic level, and how exactly this entire process was happening. One interesting thing came out, what we call it punctuated evolution. Certain mishaps were happening early on, then small mishaps were happening few years later, and then finally one more disruptive process happening and the whole chromosome got shattered. And once it got shattered, it tried to refix it back, but when it was fixing it back, it was connecting ear to the nose and finger to the thumb, and that kind of restructuring is known as chromothripsy and chromoplexy. And net result was a very abnormal DNA was being produced, and that contributed to the genesis of a more aggressive prostate cancer. And throw into this the discussion about the androgens. Androgens in prostate cancer can impact almost all of these pathways which I have talked about. The oncogenes, the suppressors, the inflammation, the angiogenesis, genomic alteration, all that can be mediated or propagated by the androgens, and that happens to be the main treatment modality what we are dealing with in prostate cancer. So this just tells you how intricately involved this entire metabolic pathway is. The only reason I can talk about it is because of understanding of little bit more about the genes. I don't want to re-go into this entire process, but I will show that growth factors of cells becoming 1 to billion is happening on the left side, but what I worry about what is happening on the right lower side, how the cancer cells are becoming from an average cell which is number increase in mass to a more invasive metastatic cancer, and this all can be quantified by different genetic tool which we have now. So that's the important part, and we finally know that in a simplified version there are few different kinds of prostate cancers. I'm not talking about the Gleason grade. I'm not talking about the stage of the cancer. I'm talking about that 71% of the cancers will have a bottom line abnormality in the androgen receptor molecules. But 49% of the patients will have something totally unrelated, which we call the Pi3 kinase pathway. Just to connect you back on what is this Pi3 kinase pathway, is insulin receptors, sugar, insulin activates a metabolic pathway inside the cell, and that is known as a Pi3 kinase AKT mTOR pathway, and ultimately it manages the certain parts of the metabolic system. That I will call it sugar addiction. So 49% of the prostate cancers have something abnormal in the Pi3 kinase pathway, which is something to do with the insulin pathway. Then 13% of the patients have an abnormality of what we call a DNA repair pathway. You remember early on I was saying that the DNA is checked for the proofreading, and if it has a minor abnormalities, the spelling mistakes, it can be corrected, and that entire process is known as a DNA repair pathway. Those DNA repair pathways are also known as the BRCA because that is very famous group of certain genes which are involved in the breast cancer discussion. But the similar gene group is involved in prostate cancer also, and 13% of the patients with prostate cancer who have an aggressive prostate cancer actually have a problem with their fixing mechanism of DNA repair. So DNA is being damaged in a normal form, but its repair mechanism is not working, and a mismanaged damaged DNA is allowed to become a living cell in the community, and that sometimes happen to carry a certain particular genomic abnormalities of the one which we just talked about and becomes a cancer. Then we talked about the cell cycle discussion. You remember that there are three different cell cycle blocks so that the cells are proofread and allowed to go to the next level only when they are good. There may be a problem in those pathways, and if that is so, 21% of the time when we are finding that there is a problem there. How does it matters? DNA repair pathway, there are certain medications which are out there. DNA repair pathway also leads to what we call a mismatch issues, or cells become a little bit more recognizable to the immune system. So mutational pathway goes up. So DNA repair pathway, if we recognize has an issue, we have certain medications built in for them. Someone has a Pi3 kinase issue and we are just depriving them of 71% of the androgen receptor. They're not going to budge. We need to understand what is happening in a molecular level to a particular patient so that we can give a more targeted treatment. Even in the androgen, there are issues with the splice variants. There are so many details which we are getting that we can tailor the treatment not for a organ, not for a tissue, but for a gene, but for a chemical, but for a target. And that is where this entire field is evolving, and this is just a good example of how understanding of the genes allows us to reclassify patients. Then comes a discussion about the genes, and there are two words I want you to understand. So far, all what I have talked about is what is known as a somatic mutation. The genes within the cancer are abnormal, rest of the body is doing fine, and those abnormal cancer genes are somatic mutations, and they took over the process and they are ruling right now. But there are certain mutations which are present right from the birth, and these are known as the germline mutations, meaning they are present in almost every cell in the body. Retinoblastoma gene you have heard about. There are Lynch syndromes in which there are certain abnormalities built in, inherited by us, which predispose us for more cancer. So that is known as a germline screening versus going deep inside the cancer which is already developing, and that's known as a somatic screening. It's very important for us to understand both are genomics. One is in all the cells, which is a germline, and other is in the somatic cells, which is happening within the cancer cells. So what are the practical utilities of this entire discussion? And I will take a couple of scenarios. I'm not taking all of the scenarios. Many a times we do the prostate biopsy and the biopsy comes back to be negative, but the patient's PSA continues to rise and we don't know how to advise them. So prior negative biopsy, we have a tool which is based on what we call an epigenetic or a halo effect of the DNA methylation project, in which tissue which is not showing cancer on a microscopic level, on a genomic level, is still showing certain changes which are not yet there for a pathologist to pick up that is a cancer, but it is showing a predisposition that in next 3 years, 5 years, this patient's tissue is likely to change into what you call cancer. If that is positive, you will have a different kind of a follow-up regimen into a patient, and at least in United States that's available as in Confirm MDX, and it's a good tool for me to use whenever we are dealing with a patient with a prior negative biopsy. We found a cancer. Cancer is a Gleason 6. or a Gleason 7 3 plus 4. What to do with that cancer? There are many different kinds of treatments which are available, and the patient could be a very adequate treatment for what we call a watchful waiting or an active surveillance. Some of them would like to have focal therapy, some of them will be a very good candidate for a nerve sparing robotic prostatectomy, and some of them needs radiation. How to kind of quantify, substratify a patient who already has a diagnosis of cancer, and that is where different other kinds of tests which have become available, and one of them is known as a Polaris, which gives a sum total of these different kinds of variables I was talking about it. It gives a risk profile for that patient as to what's the likelihood that this patient's cancer will behave more aggressively down the road. And there are more tests. One of them is known as an Oncotype DX, which has different genes which are associated with the worst outcome, and there are certain genes which are associated with the good outcome. Stromal response genes and the proliferation genes are associated with the worst outcome. Androgen signaling and cell organization genes are associated with the better outcome. So knowing what kind of genes which are there in that Gleason, same, similar looking, but very different inside, can have an impact, and we tend to use a lot more for our selection of the patients on an active surveillance. The third one is a decipher. It has multiple genes and it has 22 different kind of an RNA biomarkers which it has used. It kind of gives a risk profile as to what's the likelihood of this cancer will behave more aggressively versus less aggressively. And this can also be used not just in the biopsy tissue, but we tend to use it after a radical prostatectomy, understanding what's the likelihood that this patient's cancer will try to come back. This is all happening because of our understanding of the gene, and it gives a big picture. Polaris, OncoDX, Decipher, all of that combined or individually will help us in choosing the patients better for active surveillance. It will help us in identifying which patient may actually need what you call an adjuvant treatment, which patient can need a neoadjuvant treatment. There are newer kind of treatments which are becoming available so that the recurrence rates are lower, and that all can be tailored through this approach. Even looking at the pathology, there are certain pathways like Ki67 and the androgen receptor. You can go deep dive into an image analysis of the pathology and upgrade that with the what you call an immunohistochemistry, and you can get to the genetic level in that, and your pathology becomes an augmented pathology. It becomes a genomic guided pathology, path genomics. And I have been involved in this paper with my colleague Carlos Cardon, who is the chair of pathology here. It makes an impact in us making a decision just looking at the histopathology. So I won't just say this is a Gleason 6. We have a little bit more angle into that. And then comes a new field which is evolving, and I'm very happy that my group is quite involved. The MRI is becoming more and more sophisticated, and looking at the MRI images, looking at the biopsy images, we can combine all that and come up with what you call a better understanding how it correlates with the genomics. So we did all that. We correlated the genomics, PIRAD, and histopathology, and found that there are different pathways which are being involved in PIRAD 5 versus PIRAD 4. And in fact, there are other people who have done work and showing that certain invisible cancers have got a genomic pathway. So there may be an staying under the radar stealth mode of an prostate cancer which is using certain genes which is not allowing us to pick up using our conventional imaging methods, but genomics can unmask them, and that all is happening right now. Then there are certain racial groups which are having more prostate cancer, and we have done an analysis as to what is going on, and certain factors are emerging which are immunological, which are biological, which are inflammatory, and we are finding certain P10 losses. We talked about the P10 as in one of the tumor suppressor genes. We talked about the epigenetic analysis that talks about the methylation which may be happening more in African American. It helped me in making a better decision. It helped me in better understanding. It helped me in defining why a particular racial group is having more prostate cancer and which kind. But is it having an impact on me treating the prostate cancer? And answer is yes. Future is here because we have done couple of analyses in which we are looking at the immune checkpoint therapies and the targeted therapies. We can now know the patient has an androgen receptor kind of a problem or a neuroendocrine kind of a problem, which will have no impact from an androgen deprivation, rather will need a totally different kind of a treatment. We know which patient has an DNA repair pathway discussion, and so we can put them on the PARP inhibitors very early on, and who amongst these will be benefiting from the chemotherapy. All that can be done based on our understanding of this entire process. And also we get to know not just the cancer cells but the cells which are surrounding the cancer, what we call a tumor microenvironment. And tumor microenvironment is very rich with certain immune cells, and immune cells operate through those molecules, and these molecules are controlled by the genes. And the proteins, molecules, genes, that interaction comes here, and we can be very precise in understanding how to boost up the body's own immunity to control this. And we have written these different papers, and one of them which is we are very proud of is we are doing what we call a personalized genomic vaccine. Cancer cell, in order for it to become cancer, it sometimes hacks certain normal control mechanisms, and in that process it has some unique proteins. It brags about that it has those unique proteins, but that unique protein makes it a target because the normal cells in the body don't have that protein. Cancer cells, because of bad behavior, has developed those proteins or is using those proteins for their benefit. But we can recognize, or we can train our own body's T cells and the NK cells to go and fight only those cells who have that abnormal protein architecture. We call it neoantigens, and we have a whole vaccine trial going on in which we are developing a personalized vaccine which will work for Mr. Smith or will work for another individual patient, and it will be very unique, very target, and it is happening as we talk. We have written books on this entire topic. I'm about to put together a second edition of this whole prostate cancer comprehensive book, and that passion about understanding the biology beyond just focusing on the nerve sparing aspect, which is my full-time job. I really want to thank the organizer for allowing me to share my perspective about the genetics, genomics, and how it can have an impact in the patient care. Thank you very much.